This is Tylenol. It's the best-selling brand name for acetaminophen. This generic drug is also a component of over 600 medicines worldwide, ranging from everyday migraine meds like Excedrin to high-strength opioid combos like Vicodin. But despite how common it is, nobody actually knows how acetaminophen lowers pain. There are some leading hypotheses, but nobody's locked down the drug's mechanism of action yet. So for this video, I wanted to learn more about the origins of acetaminophen. Then I wanted to learn how it hit the market as name brand Tylenol, and then as an ingredient in so many other drugs. And finally, why its mechanism of action is still such a mystery. But we need to rewind a bit, because during my studying for this video, I learned that scientists originally thought acetaminophen would be too toxic for human use. Up until the 1800s, most effective drugs were inspired by plants and traditional remedies. Scientists used opium to isolate morphine, coca leaf to make cocaine, and the bark of the cinchona tree to make quinine. But that changed in the mid-1800s when chemists started experimenting with synthetic dyes based on coal tar. Within a few years, Germany especially had built up a bunch of expertise in coal tar chemistry and realized that some of these dyestuffs could be repurposed to treat infections. And one of those potential drugs was a coal tar byproduct called naphthalene. It was otherwise a waste product from coal processing, so it became a cheap, accessible medicine. As the story goes, one day in 1886, a patient went to his doctor to treat his intestinal worms. There, two of the doctor's assistants, Paul Hepp and Arnold Kahn, prescribed him naphthalene, and while their treatment didn't work for the worms, when the patient came back a few days later, his fever had gone away. Hepp and Kahn didn't expect that, so they looked into it. Eventually, they learned that the drug that their patient got wasn't actually naphthalene. Their pharmacist had accidentally filled their order with a compound called acetanilide, which is another byproduct of dye manufacturing. But unlike naphthalene, it wasn't really used as a drug back then. So Kahn and Hepp had this light bulb moment. Maybe this was a happy accident. Well, happy for them anyway, the patient still had worms, but maybe acetanilide would make for a good antipyretic, or fever reducer. So they ran a small clinical trial in 24 humans and confirmed that it did bring down fevers. And by coincidence, Hepp's brother happened to work at a chemical company called Cal & Company. So in 1886, Cal & Co. started producing and selling acetanilide as a medicine, what they called antifebrin. And the name was a clever branding move, because regular old acetanilide was a common byproduct of tar manufacturing. You could get it just about anywhere. But by naming it antifebrin, they give it some brand power. Unlike modern brand names though, antifebrin was cheap, only 25 cents an ounce compared to its competitor antipyrine's $1.25. Partly because acetanilide wasn't patented in the US and it was cheap to make. But it came with some severe side effects. Something about the acetanilide converted hemoglobin, the molecule in your red blood cells that allows them to carry oxygen, into methemoglobin, which doesn't transport oxygen like regular old hemoglobin does. And without oxygen, the body can develop a kind of anemia, the skin can turn blue, and in severe cases the person could die, which is a pretty bad side effect. Acetanilide's side effects became so notorious that a journalist wrote in 1905, quote, "Acetanilide, as the basis of headache powders, is prone to remove the cause of the symptoms permanently by putting a complete stop to the heart action." Which is true, you can't have a headache if you're dead, so there was room to improve on antifebrin. And that's where the Friedrich Bayer company comes in. They actually got their start back in the 1860s as a dye company. And they noticed that after making blue dye in particular, they ended up with tens of thousands of kilograms of this chemical called para-nitrophenol, or just p-nitrophenol. So knowing that acetanilide also started as a waste product, the Bayer chemists wondered if they could turn this p-nitrophenol stuff into a useful medicine. And in 1887, one of them did. They added acetyl to p-nitrophenol and eventually got a drug that Bayer called phenacetin. This one didn't lead to methemoglobin, so it sold extremely well, and ultimately kicked off the success of Bayer's pharmaceutical division. Later, chemists refined the phenacetin production process. They'd start with p-nitrophenol, then turn it into p-aminophenol, before adding acetyl to get an intermediate product called acetyl para-aminophenol. This is what we know today as acetaminophen. But it wasn't a drug yet, so scientists back then would have used its chemical name, which I'll abbreviate as APAP. A chemist at Johns Hopkins named Harmon Northrop Morse had published a method for synthesizing APAP back in 1878, but he didn't use it as a drug. Scientists waited another 15 years before someone dug it back up. And that person was Josef von Mering, a celebrity of physiology at the time. And while working with Bayer in 1893, von Mering tested APAP on humans and concluded that it would be too toxic for the same reason acetanilide was. He thought it led to methemoglobin in the blood, as well as some kidney issues. But von Mering was wrong. Something may have contaminated his sample, but APAP doesn't work like that. Still, von Mering was so influential and his results spread. So for years, doctors thought the drug was too toxic to be useful as human medicine. Either way, only 6 years later in 1899, Bayer came out with aspirin, which pulled focus from the other potential analgesics. All that's to say, acetyl-para-aminophenol, the molecule we know as acetaminophen, was discovered before aspirin. But because of von Mering's early results and his influence, the next few decades of scientists thought it was too toxic to use as human medicine. It wasn't until the 1940s that scientists started to look at APAP again. In 1946, one of the labs at the New York City Health Department was tasked with investigating the link between acetanilide and phenacetin use with methemoglobin. Basically, why are all these people turning blue after taking these drugs? They appointed a scientist named Julius Axelrod to look into it, but he had mainly studied vitamin metabolism up until then, so he would need some help if he wanted to crack the case. He reached out to Bernard Brodie, a British pharmacologist working at NYU, for guidance. Brodie was known for applying his knowledge of chemistry to drug metabolism, so their collaboration was kismet. To study acetanilide, they would give human participants 1 gram of the drug by mouth, then measure what kind of compounds were left over in their urine to see what acetanilide broke into. And in their first experiments, they confirmed what other researchers had also found, that most of the drug was metabolized into APAP. But some of the acetanilide was metabolized into aniline, another one of those coal tar derivatives. And that made them wonder if it was aniline, not APAP, that caused acetanilide users to turn blue. So Brodie and Axelrod tested each of the metabolites, APAP and aniline, separately to see if one created methemoglobin while the other didn't. To do this, they gave participants 1/10 of a gram of aniline by mouth, and in the end, they found a relationship between aniline concentration in the blood and rates of methemoglobin. More aniline in the blood, more methemoglobin. And they suspected that it was a metabolite of aniline called phenylhydroxylamine that caused problems. To test this, they gave a few milligrams of that compound to dogs and saw more methemoglobin than the other drug products. Tests since then have confirmed that phenylhydroxylamine is indeed the chemical that turned hemoglobin into methemoglobin. But that didn't automatically mean that APAP was safe. So for the same article, Brodie and Axelrod administered 1 gram of APAP to patients, and this time, when they analyzed the chemicals in their urine, they couldn't find any aniline or chemicals that might lead to methemoglobin in the blood. Brodie and Axelrod's paper came out in 1948 in the Journal of Pharmacology and Experimental Therapeutics under the epic-sounding title, The Fate of Acetanilide in Man. They concluded that aniline, not APAP, was what caused methemoglobin in the blood after all, and they proposed that APAP was the analgesic compound, the one that caused pain relief. To get more data on that analgesic effect, Brodie collaborated with a different scientist and published in the same issue of the same journal. They gave 12 female participants APAP or a placebo and measured their pain response when exposed to heat. As expected for an analgesic, the women who'd taken APAP had a higher threshold for pain than those given the placebo. This was a big deal, because after decades of thinking this compound would be too toxic to be useful as a drug, APAP turned out to be a safe and effective analgesic, which meant now it was time for the drug companies to get involved. One of the first was approved in 1950 and sold by Squibb under the name Trigesic. Tri because it contained the trio of APAP, aspirin, and caffeine. But it was taken off the market soon afterwards because of suspicions that it caused a blood disease. From there, a bunch of other companies came out with APAP products, but it was a small American pharma company called McNeil Laboratories who popularized it. At first, they combined the drug with a barbiturate, but they pivoted to a new, single-ingredient product instead. This was the birth of Tylenol. McNeil took the full chemical name, acetyl-para-aminophenol, used the TYL from acetyl and the ENOL from aminophenol, and in 1955 launched it as Tylenol. Technically Tylenol elixir since it was dissolved in alcohol. Now, you'll notice that these ads use the full chemical name. That's partly because Tylenol was initially prescription only, so these ads were meant to be read by the prescribing physician, which meant some scientific jargon was okay. But still, acetyl-para-aminophenol is a mouthful. So, according to a 2003 reference book, McNeil also gave the drug its American generic name, acetaminophen. And just like the brand name, it's a reference to the chemical name. Same thing with paracetamol, which is the generic name most of the rest of the world uses. It's another reference to the chemical name. The other thing you'll notice from these early ads is that they take direct shots at aspirin, which was the incumbent analgesic at the time. This was in the early 50s when scientists were learning more about the gastrointestinal side effects of aspirin, plus some kidney problems with phenacetin, which was still around. So McNeil's initial marketing strategy played up Tylenol as the safer, tummy-friendly alternative to aspirin. They also positioned their brand as the prescription-strength fever reducer for kids. In 1959, Johnson & Johnson bought McNeil Labs, and by the following year, Tylenol was made available over the counter, and J&J got approval for the tablet version of Tylenol. From there, the Tylenol brand name starts to spread. It got to the point where politicians and press conferences use the brand name when they should use the generic.
Don't take Tylenol. Don't take it.
Wait, sorry, that was in 2025. Beyond McNeil and Johnson & Johnson, tons of other brands started using acetaminophen in different products. The late 1970s saw some of the more infamous prescription drugs come out, like Percocet, which combined acetaminophen with oxycodone, and Vicodin, which is acetaminophen and hydrocodone, another opioid. But acetaminophen made its way into common over-the-counter drugs too. Remember Squibb's old Trigesic, acetaminophen, aspirin, and caffeine? Well, in the 1960s, Bristol-Myers, who eventually merged with Squibb, released the same trio of drugs under the brand name Excedrin. As acetaminophen got more popular throughout the 70s, it started to replace aspirin and other NSAIDs as some people's go-to pain reliever, mostly because it didn't have those same GI symptoms. But the big shift came in the early 1980s, when the CDC started looking into a connection between aspirin and a condition called Ray's syndrome, a condition that can come with intense brain and liver issues that sometimes follows viral infections like influenza. There had been a few concerning stories in the 70s pointing to a link, but after a trio of case control studies were reported in Arizona, Michigan, and Ohio starting in 1980, the CDC recommended not to give aspirin to kids with the flu or with chickenpox until they knew more. The FDA got a committee together as well, and concluded that the totality of evidence pointed towards an association. This led to June of 1982, when the Surgeon General recommended against using aspirin for kids with flu or chickenpox. And that led to even more follow-up studies. But here's what's fascinating to me. The researchers recruited dozens of hospitals with the goal of studying 100 cases of Ray's syndrome. But in the 17 months of study, after the Surgeon General's warning, they only counted 33 cases, which was much lower than they expected. And since they didn't get the expected cases, they discontinued the study. This was even more evidence for the case that aspirin use was associated with Ray's syndrome in kids. So in 1986, the FDA started requiring a warning label on products containing aspirin, and cases dropped further. This was the inflection point for acetaminophen. With all this talk about aspirin, acetaminophen seemed like a safe alternative, and brands like Tylenol got a big boost in sales. By the early 80s, acetaminophen had become the most popular over-the-counter pain reliever in the United States. But Tylenol wasn't perfect. Starting in the 1970s, reports started popping up about liver toxicity in people who'd accidentally taken too much acetaminophen. Acetaminophen has a narrow therapeutic window, which I think of as the range of dosages where you get enough of the drug to have an effect, but not too much that you do harm. And while doctors have a pretty good idea of how big doses of acetaminophen can harm the liver, it highlighted a different problem. Nobody actually knows how this drug lowers pain. Scientists finally started looking into acetaminophen's mechanism of action in the 70s, and since it was an analgesic and antipyretic like aspirin, scientists wondered if it had a similar mechanism of action, too. In 1971, a scientist named Sir John Vane released a paper where he proposed a mechanism for how acetylsalicylic acid, the active ingredient in aspirin, might work. In the paper, he proposed that aspirin inhibited the synthesis of chemicals called prostaglandins, which are chemicals involved in inflammation. Very quick biology lesson here. When something triggers inflammation in your body, your cells produce different substances which become something called arachidonic acid. Then, enzymes called cyclooxygenase enzymes, or COX enzymes for short, tweak it into prostaglandin H2. From there, it can become different prostaglandins and lead to fever and inflammation, or it can become thromboxane and encourage platelets to clump together. Vane's research showed that something about aspirin inhibited prostaglandin formation, probably by inhibiting the COX enzymes that converted arachidonic acid into prostaglandin. And that would explain how aspirin could reduce inflammation, lower fever, and work as a blood thinner all at the same time. By inhibiting COX enzymes, you inhibit everything prostaglandins lead to. Vane's work led him to win a share of the Nobel Prize for Medicine or Physiology. But before winning the prize, he and a colleague named Roger Flower tried to explain acetaminophen's mechanism of action too. The researchers were British, so they called it paracetamol, but I'm going to call it acetaminophen just so I'm not switching back and forth for the next few minutes. Vane and Flower knew that acetaminophen lowered pain and fever like aspirin, so they thought it might work the same way, by inhibiting COX enzymes. But unlike aspirin, acetaminophen has essentially no anti-inflammatory activity, so it couldn't work through exactly the same pathway. Now, at the time, the researchers imagined that anti-inflammatories worked locally, meaning at the site of inflammation. So if you sprained your ankle, ibuprofen inhibits COX enzymes from making prostaglandins at the ankle. But around the time, groups of different scientists were making breakthroughs with cerebrospinal fluid, which is an important fluid in the brain and spinal cord. And throughout all their experiments, they'd made a case that prostaglandin-like substances existed in the central nervous system, which is the brain and spinal cord. They also showed that acetaminophen could lower the concentration of these prostaglandin-like chemicals during fevers. And that hinted that acetaminophen might work centrally rather than peripherally at the site of inflammation. Just to be clear, centrally in this context refers to the central nervous system, while peripheral means everywhere else that isn't the brain or spinal cord. So Vane and Flower tested the idea. They took brain tissue samples from rabbit to represent the central nervous system, and spleen tissue from dogs to represent peripheral tissue. Then they exposed each tissue to aspirin, acetaminophen, or an anti-inflammatory called indomethacin. From there, they measured how well each drug inhibited prostaglandin formation in each tissue type. They found that aspirin inhibited prostaglandin production well in both types of tissues, while acetaminophen was much more potent at blocking prostaglandin formation in brain tissue than in spleen tissue. They published their paper in 1972, and it became the strongest evidence that acetaminophen worked differently than aspirin. Not at the site of inflammation, but through the central nervous system. But in science, every new answer begets new questions. See, when other researchers tested this central mechanism for acetaminophen, they got weird results. In early studies, neither COX-1 nor COX-2 enzymes responded to acetaminophen at the doses you'd see in most medicines, and this made scientists rethink whether the drug actually inhibited COX enzymes. In 2002, a research group from Brigham Young University published a paper in the Proceedings of the National Academy of Sciences. They noticed that under specific genetic conditions, the gene that codes for the COX-1 enzymes could lead to distinct new enzymes. They called one of those enzymes COX-3, although sometimes it's referred to as COX-1b. They found this COX-3 enzyme in brain tissues from dogs, and knowing that acetaminophen probably worked on the central nervous system, they wondered if it maybe inhibited this enzyme. So, they took arachidonic acid at different concentrations and exposed it to the COX-1, 2, and 3 enzymes, which ought to turn it into prostaglandin. Then, they tested different anti-inflammatory and analgesic drugs on them to see how they disrupted that process. Sure enough, acetaminophen inhibited COX-3 from completing its reaction. They also brought some medical history into their experiment and tested old-school phenacetin, and it turned out to be a more potent inhibitor of COX-3 than even acetaminophen. As a result, they concluded, quote, "Thus, inhibition of COX-3 in brain and the spinal cord may be the long-sought-after mechanism of action of acetaminophen." But this theory had some issues too. The researchers from the 2002 paper got their dog brain tissues to make COX-3 under some pretty specific genetic conditions, and other researchers doubted that COX-3 enzyme could be made in human brain tissue in a meaningful amount, at least not in an amount that would explain acetaminophen's pain relief effects. At the same time, other research groups have found functional COX-3 in human cells, although even that group doubted it explained the drug's analgesic effect. Beyond that paper, other research groups wondered if acetaminophen might work by selectively inhibiting COX-2 more than COX-1, but that probably wasn't it either. Drugs that selectively inhibit the COX-2 enzymes are strong anti-inflammatories, and acetaminophen isn't. Plus, COX-2 inhibitors have their own problems. I'll get around to making a video about the Vioxx scandal someday. There was one more possible COX-related mechanism. In order to kickstart the process of prostaglandin formation, COX enzymes need this little free radical called tyrosine 385 to get oxidized, meaning tyrosine loses electrons. Researchers have proposed that acetaminophen acts as a reducing agent, meaning it adds electrons, which is the opposite. So instead of thinking of acetaminophen as blocking the COX enzyme's binding site, this hypothesis says that acetaminophen prevents COX enzyme from becoming active in the first place. To summarize up to this point, scientists started out thinking that acetaminophen inhibited COX-1 and 2, some thought that it was one or the other, and some thought that it might be COX-3. But COX-3 might not be all that relevant in a real human body. The biggest lesson learned, though, was that acetaminophen probably acts centrally rather than in the periphery. While scientists were looking into the COX enzymes, other scientists were testing a totally different mechanism of action in the central nervous system, one that focused on pain signaling. When neurons send pain signals from the periphery to our brain, those signals pass through a region of the spinal cord called the dorsal horn, dorsal referring to the backside. These pain signals are processed there before heading up to the brain where they're perceived as pain. Now, there are projections that pop off from the brainstem that go down the spinal cord and enter the dorsal horn of the spinal cord too. These projections pump out serotonin. In this case, serotonin acts as a chemical messenger which may inhibit pain signals back up to the brain. The idea got more popular after a team of Norwegian scientists published a rat study in 1991. They used a special chemical to lesion, or damage, that pathway from the brain to the dorsal horn in some of the rats. Then, they'd take the rats, inject one of their paws with a little bit of formalin, and categorize their response as normal behavior or pain-related behavior. The researchers wanted to see if acetaminophen would change the rat's behavior. They noticed that when they gave acetaminophen to rats with lesioned neurons, the effect of the drug wasn't as strong, suggesting that acetaminophen used some part of that pathway to reduce pain. But the details were shaky, so other researchers wondered if serotonin receptors were involved. In 2006, a French research group tested this. They took human participants, gave them little electrical shocks, and measured their pain. Then they did the same thing, but gave them acetaminophen, which inhibited pain as expected. But then repeated that process while giving them drugs that blocked the serotonin receptor. And this time, the acetaminophen didn't reduce the pain from the electrical shocks. The authors of that paper concluded that the stimulation of the serotonin receptor was somehow involved in acetaminophen's pain-relieving mechanism. Fast forwarding a bit, it's not that acetaminophen directly stimulates serotonin receptors. Instead, it somehow enhances the body's natural serotonin pain control pathways, possibly by acting on specific serotonin receptors in the spinal cord called 5-HT receptors. But this model opens up a few other questions. Like, the chemical that the French researchers used to block serotonin receptors also blocks a few other receptors, so there's a chance that the other ones it blocks could be involved too. But all of these explanations have had a big update, because in 2005, a team of Swedish researchers identified a new metabolite of acetaminophen. They showed that when acetaminophen is metabolized by the liver, it forms para-aminophenol, which crosses the blood-brain barrier. Then it combines with arachidonic acid to form a new substance called AM404 for short. The Swedish researchers propose that AM404 is the active metabolite of acetaminophen, meaning this is the chemical that might lead to pain relief. And there are a few routes it might work through. Scientists learned that AM404 inhibited the transport of a chemical called anandamide, causing it to build up. And that buildup stimulates a type of receptor called endocannabinoid receptors. As you could infer from the name, these are the receptors for cannabinoids, like the chemicals in marijuana. But they have a bunch of other roles in the body too, like in pain modulation. You've got these cannabinoid, or CB receptors for short, throughout the nervous system, both in the brain and on pain-sensing neurons. And just like the serotonin receptors I mentioned a minute ago, when those are stimulated, especially a receptor called CB1, they send a signal from the brain down through the spinal cord and tamp down pain signals. And when researchers blocked those CB1 receptors in rats, acetaminophen didn't relieve pain as well, which was evidence that these receptors were involved. But this AM404 metabolite might work through another receptor called TRPV1, a receptor that usually leads to the sensation of heat and pain. When researchers studied rats who were genetically modified to not have the TRPV1 receptor, acetaminophen didn't affect their pain response, which suggested that the drug needed this receptor to work. How exactly this mechanism leads to pain relief is a part of the big acetaminophen mystery. Like, I read sources claiming that AM404 is either an agonist, a stimulator, or an antagonist, a blocker for this receptor. This might be because of downstream effects or sensitivity of the receptor to AM404, or maybe even different TRPV1 receptors working differently depending on where they are in the body. And that's kind of unsatisfying, right? Scientists have all these ideas that might explain how acetaminophen reduces pain, but nobody's locked it down. So, to summarize, for a long time, scientists used to think that acetaminophen worked the same as aspirin. They thought it inhibited prostaglandin formation by inhibiting COX enzymes. But these days, scientists think it works through multiple paths simultaneously in the central nervous system, including the COX enzymes, the endocannabinoid and serotonergic pathways, and all probably because of its metabolite AM404. But let's pull back and get some big picture takeaways. First, we didn't start using acetaminophen for such a long time because of von Mehring's influence. Von Mehring was a well-respected scientist, so even though he'd done just one experiment, when he said not to use acetaminophen, his words had outsized impact. Granted, he was also the first to test the drug clinically, so there wasn't evidence to refute him either. But the trust in his authority kept people on acetanilide for decades and discouraged folks from studying acetaminophen. So the takeaway for modern day is, before drawing conclusions, especially ones that will affect millions of people, it's more appropriate to take in the total body of research, even when it's in opposition to a celebrity scientist. Or just a celebrity. Takeaway number 2 is the uncertainty part. The fact that nobody has locked down acetaminophen's mechanism this long after discovering the compound doesn't come from lack of trying, it comes from science working as it should. Old hypotheses need to stand up to scrutiny, and when they don't, scientists propose new hypotheses and test them and learn from them. Science isn't a product, it's a process, one that gets us closer to an accurate view of the world. Speaking of science as a process, I've got a documentary recommendation for you. It's called Red Atoms, a docuseries from real-time history that tells the story of the Soviet nuclear program during the Cold War and all the challenges that went into developing atomic technology. I was somewhat familiar with the nuke building, mutually assured destruction stuff, but this series goes way more in depth into Soviet nuclear energy production, space travel, Chernobyl, and all the fallout. Soviet-era science was notoriously secretive, so footage is hard to find, but Red Atoms found some great archival clips to tie the story together visually. You can watch the entire series for free right now with a free trial to Nebula, an indie streaming service made by creators for creators. I post all my videos there early and ad-free, but Nebula is also home to hundreds of other creators, including some of your favorites in the health space like Chubby Emu at Midlife Crisis, and we're adding new folks all the time. Plus, we're always coming out with new Nebula originals, which are shows that you can only watch on Nebula. Like Adam Chase from Jetlag has this docuseries called Scav, which tells the story of this massive scavenger hunt at the University of Chicago. The first episode in the series is out now, and if you like Jetlag, you'll like Scav. It's a similar vibe of organized chaos. And with my link, you'll get to try Nebula for free for 3 days. And seriously, take advantage of it. Watch as much Nebula as you possibly can. I dare you to watch all the originals in 3 days. So if you want to check out Nebula, go to go.nebula.tv/PatrickKelly to learn more. If you use my link this month, December 2025, then after the 3-day free trial, you'll get 50% off an annual subscription, which brings it to just $30 for a full year. Or you can get a lifetime subscription, which brings the cost from $500 down to $300 for life. That subscription directly supports my work, as well as the production of interesting new content. Again, that's go.nebula.tv/PatrickKelly, or you can scan this QR code. Thanks for watching, y'all. Take care.